Yes, Dr. Pal, please start.
Good morning to all. I extend a warm and heartfelt welcome to each and every one of you to today's enlightening webinar. I am Dr. Payal Mittal, the co-convener of this event, and it fills me with immense pleasure to witness such a diverse and engaged audience gathered virtually. Your presence here not only enhances the vibrancy of our discussions, but also underscores our collective commitment to advancing knowledge in our field. I am genuinely excited about the prospect of delving into insightful and collaborative conversations as we embark on this intellectual journey together. By consistently delivering captative webinars, SPSR has solidified its positions as a premier platform for knowledge exchange and professional development in pharmaceutical sciences. I am confident that today's session will continue this tradition, offering attendees invaluable insights from our esteemed speakers. Let us take this opportunity to learn, engage, and contribute to the advancement of our field. Welcome once again, and I anticipate a rewarding webinar experience for all. Thank you all for joining us today. Today is a significant milestone as we gather for the 52nd SPSR International Webinar on Nature's Pharmacy, Tropical Plant-Based Therapies for Skin Disease Management. This event is part of a distinguished series hosted by the Society of Pharmaceutical Sciences and Research, SPSR, under the esteemed mentorship of Dr. Shashi Alok, National President of SPSR. Under his guidance, the SPSR continues to play a pivotal role in advancing knowledge and innovation in the field of pharmaceutical sciences. The SPSR is dedicated to advancing the pharmacy profession through a multifaceted approach. Our objectives encompass the organization of conferences to campaign the causes of pharmacy, contributing to programs and services that address the health needs of India and focus on disease prevention. Additionally, we actively recognize and honor distinguished pharmacists who contribute significantly to the encouragement of the pharmacy profession. At the core of our mission is the promotion and facilitation of acquisition, dissemination, and exchange of pharmacy knowledge, ensuring a robust flow of information about the practice of pharmacy and pharmaceutical services. Through these objectives, we aim to elevate the standards and the impact of pharmacy profession, fostering a dynamic and collaborative environment. We are truly honored to welcome our esteemed speaker, Dr. Yik-Ling Chew, Associate Professor and Head of Postgraduate Studies, Faculty of Pharmaceutical Sciences, UCSI University, Kuala Lumpur, Malaysia. Dr. Yik-Ling Chew brings invaluable expertise to our event, and we eagerly anticipate gaining insights from her wealth of knowledge in this field. Her presence enriches our discussions and adds significant value to our exploration of the latest advancements in tropical-based, plant-based therapies. We are grateful for her participation and look forward for a truly enlightening session. In our esteemed gathering, we are honored to have the distinguished presence of Mrs. Monika Sabharwal, the convener of this webinar, National Secretary, Society of Pharmaceutical Sciences and Research, and Managing Director, Editor of IGSPR and IJP. Her multifaceted contributions and leaderships in these capacities underscore her commitment to the advancing field of pharmaceutical sciences and research. We are privileged to have Mrs. Monika Sabharwal with us today, guiding our webinar with her expertise and dedication. Without further delay, I have the distinct pleasure of inviting Mrs. Monika Sabharwal to extend a warm welcome to our esteemed guest speaker, Dr. Yik-Ling Chew.
Thank you so much, Dr. Payal Mittal. Hello, everyone. It is my great pleasure to welcome you all to the 52nd SPSR International Webinar on Nature's Pharmacy, Tropical Plant-Based Therapies for Skin Disease Management. I, Monika Sabharwal, honored to serve as a convener for today's event, organized by Society of Pharmaceutical Sciences and Research SPSR. I am pleased to announce that we have received an overwhelming response with more than 3,500 registrations from over 23 countries. This remarkable turnout underscores the global interest in exploring the potential of tropical plant-based therapies and dedication of our international community to advancing the pharmaceutical sciences. Today, we have privilege from hearing of our esteemed speaker, Dr. Chew, Associate Professor and Head of Postgraduate Studies at Faculty of Pharmaceutical Sciences, UCSI University, Kuala Lumpur, Malaysia. Dr. Chew is a distinguished researcher whose work in the phytochemistry and phytomedicine has significantly advanced our understanding of medicinal properties of tropical plants. Her insights will undoubtedly enrich our knowledge and inspire new avenues of research and application. At SPSR, we are committed to continuously improving the standards of our webinars. We strive to bring you the best and most relevant content by engaging with the renowned speakers from around the world. Today's speaker from Malaysia exemplifies our commitment to international collaboration, and we are grateful for Dr. Chew's participation in the pharmaceutical sciences. Our journey to this 52nd SPSR webinar has been marked by the unwavering support from the participants across the globe. We deeply value your feedback, which is instrumental in shaping the topics and selecting the speakers that resonate with your interest and the professional needs. Your positive feedback received through emails and other channels not only boosts our morale, but also drives us to work harder and to uplift the pharmacy profession overall to all of us. Today, as we celebrate this milestone in our webinar series, we reflect on the immense global support and collaboration that have brought us here. SPSR is dedicated to fostering a vibrant community of pharmaceutical professionals committed to the excellence and innovation. We are deeply honored to welcome Dr. Shashi Alok, the esteemed National President of SPSR. His forward thinking and leadership has been a crucial role in spearheading a significant transformation within SPSR, making it a paragon of excellence in the pharmaceutical sciences. I would also like to extend my heartfelt gratitude to the Dr. Payal Mittal, the co-convener of this webinar, as well as the organizing committee members, Dr. Pradeep Gulani, Dr. Padmani Shukla, Mrs. Swati Jain, Dr. Saurabh Kose, Dr. Sanjay Nagdev, Mr. Angur Agarwal, and Mr. Vinay Tantre. Their unwavering dedication and hard work has been instrumental in bringing this event into a reality. Behind the scene, an outstanding team has been cornerstone of this webinar's smooth organization. Their relentless dedication and commitment to the excellence have driven the success of the event. I sincerely thank each and every team member of SPSR for their meticulous attention to the detail and unwavering passion, which have been a pivotal role. I extend my deepest gratitude to each one of your enthusiastic participation and valuable contribution to this webinar. Your engagement fuels the collective pursuit of excellence in the field of pharmaceutical sciences. I'm certain that today's discussion will be both enlightening and inspiring, leaving a lasting impact on our professional journey. Once again, I extend my sincere gratitude for each one of you for being the integral part of the 52nd SPSR International Webinar on Nature's Pharmacy, Tropical Plant-Based Therapies for the Skin Disease Management. Your active participation and engagement are invaluable contribution to our collective pursuit of knowledge and the progress in the field of pharmaceutical sciences. Stay at forefront of innovation with SPSR. By subscribing to our YouTube channel, you gain exclusive access to upcoming webinar, expert insights, and groundbreaking research in the field of pharmaceutical sciences. Join our vibrant community of knowledge seekers and actively engage in the thought-provoking discussion that will shape the future of healthcare and the pharmaceutical sciences. Do not miss out the latest updates. Remember to subscribe our SPSI YouTube channel and hit the notification bell icon for the instant alerts. Join us on the journey of discovery and innovation as we work towards advancing the pharmaceutical sciences for the betterment of society overall. Without further ado, let us welcome Dr. Chew to begin today's enlightening session on nature's pharmacy, tropical plant-based therapies for skin disease management. Thank you for being here. I hope you will like today's webinar, both inspiring and informative. Over to you, Dr. Payal Mittal.
Thank you, Mrs. Monika Sabharwal, for allowing me to conduct this webinar. And now let's begin with a brief introduction of today's speaker. Associate Professor Dr. Chew Yik-Ling is a distinguished researcher and educator at UCSI, University's Faculty of Pharmaceutical Sciences. She holds a BSc Honours in Biotechnology and a PhD from Monash University, along with a postgraduate diploma in tertiary teaching from UCSI University. Additionally, she is a chartered scientist and a member of Royal Society of Biology in the UK, reflecting her dedication to scientific rigor and professional excellence. Dr. Chew's research is renowned for its in-depth exploration of phytochemical properties and medicinal potential of tropical plants. Her extensive studies on the antioxidant, anti-inflammatory, and antimicrobial activities of these plants have significantly advanced the field of phytomedicine. These properties are crucial as antioxidants help to protect cells from damage, anti-inflammatory agents reduce inflammation, and antimicrobials combat infections. Her research not only enhances scientific understanding, but also contributes to the development of national therapeutic agents that can be used in modern medicine. A notable highlight of Dr. Chew's illustrious career is her groundbreaking research on the effects of tropical plants and herbal medicines in managing skin disorders. She has identified numerous plant-derived compounds with potential therapeutic benefits for skin health, offering alternative treatments for various dermatological conditions. Her studies have demonstrated that these natural compounds can be effective in reducing inflammation, accelerating wound healing, and providing antimicrobial protection, thereby promoting overall skin health. Dr. Chew's contributions have been instrumental in promoting the integration of herbal medicine into modern skincare practices. By demonstrating the efficacy of natural remedies in improving skin health and treating disorders, she has paved the way for the development of new skincare products that harness the power of nature. This integration has significant implications, offering consumers safer and potentially more effective alternatives to synthetic chemicals commonly found in skincare products. Beyond her research, Dr. Chew is an influential educator and mentor. She plays a critical role in shaping the next generation of scientists and healthcare professionals. Her commitment to education is evident in her dedication to teaching and mentoring students, helping them to develop the skills and knowledge necessary to succeed in the field of pharmaceutical sciences. She has supervised numerous undergraduate and postgraduate research projects, many of which have resulted in significant contributions to the field. Dr. Chew's dedication to her field and her innovative approach continue to inspire advancements in phytochemistry and phytomedicine. Her work not only enriches scientific understanding, but also has practical implications for the development of alternative therapies and the promotion of natural health solutions. As a leading figure in her field, Dr. Chew's research and educational endeavors continues to influence and shape the future of phytomedicine. She remains a beacon of excellence, driving forward the integration of natural compounds into modern medical and therapeutic practices. It brings us immense joy and pride to share that we have garnered the participation of over 3,500 registered attendees from across the globe, representing a diverse array of countries. Among them, we have a significant presence from India, Albania, Bulgaria, Canada, Ethiopia, Ghana, Indonesia, Iraq, Jordan, Kenya, KSA, Kuwait, Malaysia, Mexico, Nepal, Nigeria, Pakistan, Saudi Arabia, South Africa, Syria, Turkey, UAE, UK, and USA. The overwhelming response from such a wide-reaching and varied audience is truly humbling. We extend our heartfelt gratitude to each and every participant for choosing to be the part of this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Chew Yik-Ling, with us. Welcome, ma'am.
Thank you, Chairperson.
I kindly request you to share your power presentation, ma'am, and the talk.
All right. Okay, can you see my slides?
Yes, it's visible, ma'am.
Okay, so a very good morning to some of the audience from a different part of the world, and a very good afternoon from some of the audience from another part of the Asian country. So I'm Associate Professor Dr. Chew, who is from UCSI University. Basically, my research interest is on the usage of the topical plants in managing skin disease. Okay, so just give you a little bit on the impact of the skin disorder. So basically, skin disorder happens in many parts of the world, regardless that whether you are an underdeveloped country, you are a developing country, or even you are a developed country. So basically, this is a global disease burden, and it can affect people from different ages and different culture, like from infants until old age people. Okay, and then regardless that which culture that you are from, you have the chances of having these skin disorders. And skin disorder, why it became the focus of my research is because that what I noticed is skin disorder is quite common among the Asian countries. So based on the statistics that we have seen, that it is about 1.79% of the global burden disease, which means that it will impact the socioeconomic status of the different countries, regardless whether you are from which part of the world. Okay, and then this is actually a fourth leading cause of non-fatal disease burden and the disability in 2013. Okay, so when we look at Asian country, there is a very high burden of inflammatory dermatosis, which has already been reported every year, and this kind of inflammatory dermatosis skin diseases are mainly associated with itching, like example, psoriasis, contact dermatitis, atopic dermatitis, pruritus. These are among the examples which actually been reported commonly in Asia countries. So when we look at the atopic dermatitis statistics in Asia in the past few decades, we notice that the prevalence of atopic dermatitis is actually getting higher and higher every year. This could be due to the reason of urbanization, okay, and also that the increase of family income, okay, and also we are actually more having a higher exposure to allergen, like example, pollutants from the air, pollutants from the environment, Okay, so that is the chances of how the people in Asia are having higher prevalence of atopic dermatitis. And also not to forget that nowadays, almost every one of us, we have the frequent usage of bathing and soap usage for the hygiene purpose, compared to those olden days that probably that we are not using this kind of a cleansing agent so much. But now I think that every family household will have this kind of cleansing agents. Okay, so the common pattern of therapy towards the skin disorder, basically, it can be divided into prescription and non-prescription. Back in year 2017 and 2018, actually, one of my students has actually conducted a study to study on the pattern of therapy for people in Malaysia which have skin diseases. So based on the study, we have actually categorized into two major classes. The prescription, including these are the common one which has been used in Malaysia, like the topical corticosteroid, betamethasone, corbetazole, mometasone. These are among the top ones. Okay, there are other prescription medicine which is already being recommended by physician. Whereas for the non-prescription, it is very surprising that we notice that there are people who actually more prone towards using non-prescription therapy. Like example, the top one will be always the topical calamine and the white soft paraffin. Okay. Other than that, we also notice that there are patients who are actually moving towards the plant-based products. Like example, using the shea butter, using the aloe vera, calendula, papaya, dexapethanol, pearl powder, or even the Cordonopis pilosula roots. Okay, so these are the non-prescription common one which has already been used. Okay, so I would like to show you that based on the study that we have conducted, you can see that these are among the products which has already been used by the people that is suffering from skin diseases. Okay, so you can see that most of the route of administrative of the therapy is through topical. Okay, and then there are different kinds of product which is you probably have seen it in the market. Okay, like example, some famous brands of the dermatological product. Okay, not to mention any of the product name over here. And just to want to highlight to you that the active ingredient of this kind of product is actually a plant-based ingredient. Okay, so these are the plant-based ingredients that people are commonly used. Okay, it looks very similar to many of us. Okay, probably when you are using some of the products over here, you may not be aware that the active ingredient which is present in the product is actually this kind of ingredient. Okay, so today, I just want to highlight 3 specific plants that is quite commonly used in Malaysia or even in some part of the Asia country. Not to say that only these 3 plant works. There are a list of plants that I have in my research databases that is actually very effective towards skin disorder. But since I have the time limitation, so that's why I wanted to focus only on these 3 plants. Okay, just to give you everyone here an overview of how these 3 plants actually works on managing the skin disorders. So by looking at the target to manage this skin disease, basically there are 4 major targets that we are looking for. One is by inflammation. We are trying to suppress the inflammation of the skin. Secondly is to combat the free radicals, which is potentially present on the skin, which actually can cause adverse reaction in the skin disorders. And other than that, we also look at the skin infection, which actually being caused by the microbial, okay, especially the bacteria and the fungus. And lastly, because of the skin disorder, like what I mentioned, itching is a very common symptoms. So when we are having itchiness, the potential of scratching your skin, it happens all the time. So when excessive scratching happens, you will have the open wound. So this is why that when we are looking for the target for the bioactive compounds from the plant, basically we will look into these 4 main types of activity. So therefore, my students basically, when they're looking at the managing skin disorder, we will focus on 4 different activities, including anti-inflammatory, antimicrobial, antioxidant, and wound healing. Okay, so first of all, let's go to the first plant that I wanted to talk about today, which is on the coconut oil. Okay, so for your information, coconut oil, the scientific name is called Coccus nucifera. Okay, I believe that some of you, you have a coconut oil at home that you use for cooking, goes for baking, which is actually very common ingredient which is present at home. But from our study, basically, we actually found that coconut oil is actually very good in targeting the inflammation which actually happen on skin. Okay, because coconut oil, they do have some ingredients which can actually manage to inhibit some of the inflammatory pathway which is actually present in the skin inflammation. This is due to the presence of the medium chain fatty acids in the coconut oil, which actually the presence of the skin microflora will be able to convert this medium chain fatty acids into a free fatty acid form, which is beneficial to the skin. Okay, like example here, we have the example of like capric acid, lauric acid. Okay, so these are the 3 examples of medium chain fatty acid which is present in coconut oil, but there are actually more than this, what is reported here today. Okay, so the interesting part is what we notice is that the microflora on your skin will be able to break down the fatty acids which is present in the coconut oil to become the free fatty acid, and this free fatty acid will be able to handle the various type of activities like including antioxidant, anti-inflammatory, and antimicrobial. Okay, so this is why that we found that why it is so interesting to use coconut oil for the skin management. Other than that, we also noticed that there is a 70% of eczema patient. Okay, basically, on their skin, they are colonized with Staphylococcus aureus. So I would say that those patients with the eczema or atopic dermatitis, they have quite commonly being colonized by the Staphylococcus aureus. However, after the application of the coconut oil on their skin, it was found that after 21 days, there is an inhibition of 95% of the S. aureus on the skin. Okay, that means that from 100%, it was initially present. After 21 days of treatment, the colony of S. aureus has been reduced up to 95%. Okay, this is one of the interesting part that we are very convincing that coconut oil has the capability of inhibit the S. aureus which is present in the skin. Other than that, we also noticed that the time for wound closure and the time for complete epithelialization on the people with dermatitis issue, that the time actually taken for the wound to close is actually much lower compared to those patients who is not using coconut oil as a complementary therapy. Okay, and other than that, we also noticed that the amount of pepsin soluble collagen and the collagen cross-linking and the collagen content which is present on the skin is actually increasing upon the application of the coconut oil as a treatment in the atopic dermatitis. Okay, second plant, I'm not too sure that how many of you actually have seen this plant. This is called the candlebush. Okay, it is quite common in Asia countries. Okay, so the scientific name of this plant is called the Cassia alata. So Cassia alata, basically, they have this very distinctive orange, yellowy kind of a flower. Okay, this is commonly, you can find it in wild. You don't see it very commonly in those gardens because basically this plant is a plant which actually grow in wild. But there are a lot of reporting activities on the usage of this plant on different kind of diseases, and one of it is on skin disorders. So what through the literature, we actually found out that the roots, bark, stem, and the leaf of this candlebush has a moderate antimicrobial activity towards S. aureus, and also true enough that we actually did some of the study on the antimicrobial activity in the lab, and we do found that it consists of a moderate antimicrobial activity to S. aureus, Okay, and when we try to research further, we notice that the presence of different kind of phytochemicals in this plant is actually playing a very major role in exhibiting the activity. Examples like in the literature, there is reported the camphorol, aloimodine, astragalin, okay, and also other camphorol derivatives. And other than that, we also notice a presence of the gallic acid, caffeic acid, and also not to forget that the presence of the major compound, which is rain, from this plant, and also different kinds of fatty acid is also present inside this plant. Okay, so based on the study that we have conducted, we found that the phytochemicals which is actually present in the candle bush, they are able to target on the bacterial surface in order to achieve the antibacterial activity. So what the compound actually did was to attach to the bacterial surface, and this compound will affect the membrane fluidity of the bacterial cell wall. So when the bacterial cell wall fluidity has already been affected, we notice that the bacterial cell, the cell wall, it looks a little bit different compared to the untreated one. Like example here, you can see that the untreated one is very nice, circular, okay, very smooth surface. Whereas after the treatment, we notice that the bacterial cell actually has a little bit like wrinkle kind of appearance. And this actually signifies that the membrane fluidity of the bacteria has already been affected. So because of the fluidity has already been altered, we notice that the physiochemical properties of the bacteria may be changing also, and that actually cause the bacteria to die due to potassium leakage, and therefore slowly it will eventually cause a cell death. So when we try to look at the comparison of the S. aureus before and after treatment, and we are trying to elucidate that what are the specific changes which actually happen on the bacterial cell wall, we notice that there is a changes mainly on the bacterial cell wall. Okay, not so much on the nucleic acid part. Okay, we notice that changes is actually happening in the phospholipid bilayer of the bacterial cell wall. Okay, and then there are some protein carbohydrate changes which are actually being signified in the FTIR that we notice. That you can see that if the FTIR in blue color, these are the untreated one. The one which is present in red color, these are the changes after the treatment. So when we try to put the untreated and treated cell onto the FTIR and we try to overlay, we notice that there is a changes on the macromolecular composition on this treated and untreated bacteria. So that is how we actually made out an assumption that the phytochemicals in Cassia alata is actually affecting the bacterial cell membrane and causing them to die. Okay, other than that, there is a presence of other phytochemicals in Cassia alata, like example, caffeic acid. Okay, it's already been having a lot of studies which actually reported that this caffeic acid has a very broad spectrum of antibacterial activity, which means that it is able to inhibit the gram-positive and gram-negative bacteria. So how this caffeic acid affects or exhibit the antibacterial activity is by affecting the functional peptide proteins and ion channel, which is present on the membrane of the bacterial cell. And because the functioning has already been affected, and this will eventually disrupt the cell wall of the bacterial cell. So when the cell wall disrupted, they will eventually alter the bacterial cell membrane integrity, and that causes the bacteria slowly eventually die. Other than that, like what I mentioned, Cassia alata, we have a major compound which actually called rain. Rain is actually a substrate for many transporters. This rain is able to regulate the expression of different kind of transporter gene, and this is able to affect the bacterial biological mechanism, like example, that they can actually regulate the ferrochrome, cytophore, and also the heme transport, which actually can cause the cell from being suppressing from proliferation. So the cell will not be able to divide and grow anymore. And other than that, rain is also inhibiting the aerobic and anaerobic respiration of the bacteria. And eventually, because of this combination of activity, the metabolism of amino acid, detoxification, and pathogenic factor will also be inhibited upon the treatment by the rain. Okay, and also we actually have conducted, one of my master student has actually conducted the in vitro wound healing activity using the candlebush. Okay, we actually noticed that candlebush has a satisfactory in vitro wound healing activity when we try to compare with the positive control that we use in our study. So this is actually unpublished data that we wanted to share with everyone here. So allantoin is actually very commonly being used as an agent to improve wound healing in a lot of the topical products. So when we notice that we are trying to treat the cells with candlebush extract and also with allantoin, we notice that at the same concentration, that the candlebush extract actually exhibited a much better activity compared to allantoin. And other than that, we also notice that the presence of oleic acid and linoleic acid is very effective to us in modulating the inflammatory response for tissue repair and wound closure. They also inhibit the accumulation of leukocyte at the skin lesion, okay, and also that will inhibit the cationic serine protease activity in wound. And part of the activity which is exhibited by oleic acid and linoleic acid is they are able to regulate the expression of MMP and tissue inhibitor MMP that will eventually promote the wound closure in tissue remodeling process. So what we have found in the literature is there is a 88.9% of reduction in wound surface area after 21 days of treatment using the candlebush extract. Okay, so when look at the anti-inflammatory activity of candlebush, we notice that one of the very common activity which is being reported on the anti-inflammatory from this plant is called astragalin. So this is also a camphor derivative. So there are a lot of literatures and studies has already been conducted by using astragalin as the single compound. And based on the literature, we found that astragalin is able to regulate the anti-inflammatory related transcription factor enzymes and cytokine. Like example, those are the common cytokine that you very often hear of is like TNF alpha, inducible nitric oxide synthase, okay, COX-2, prostaglandin E2, MMP1, MMP3, interleukin-1 beta, IL-4, IL-6, IL-8, IL-13, and also the interferon gamma. So we noticed that based on the literature, these are the common cytokines which is actually being regulated by astragalin, okay, in order to achieve the anti-inflammatory activity. Okay, we also noticed that there are various types of polyphenols which actually present in candlebush. Like example here, which I mentioned before, is like gallic acid, camphic acid, camphorol, camphor derivatives, quercetin, luteolin, epigenin, naringenin, rain, imodin, aloimodin. So why do I want to stress on the presence of these polyphenols here? Is because this polyphenol is very common, which is actually present in plant, and they actually exhibit the antioxidant activity in the candlebush. So the antioxidant activity is able to protect the skin against oxidative stress. Okay, so what we notice that if there is an administrative of the polyphenols from the candlebush, there is a significant improvement on the skin condition throughout the 12 weeks of treatment. Sample like in terms of skin inflammation, after 12 weeks, we noticed that the skin inflammation has improved 24% better. The skin dryness has improved 30% better. And also the skin texture is actually 80% better. Okay, so these are through the 12 weeks of skin treatment that we notice that the presence of antioxidants in the improvement of the skin condition is equally important. Okay, the last one that I want to talk about is on the Gotukola. Okay, Gotukola, some of the country you call it Asiatic pennywolf. Okay, but the scientific name of this plant is called Centella asiatica. So based on many literatures, it is reported that Centella asiatica or Gotukola has four core bioactive compounds They are acetococyte, metacalcitocyte, acetic acid, and metacalcic acid. So these are the core ingredients which actually present in gotu kola. So this core ingredient, we found that there is a significant increase in hydration of the stratum corneum. So there is a study which is actually reported back in 2016, which actually this researcher investigated on the usage of the product with gotu kola, with product without gotu kola. They actually found that in the product emulsion with 5% of gotu kola, there is a 25% increase in hydration of the stratum corneum. Whereas even though that they are using a different delivery method using hydrogel, with 5% of gotu kola, they also managed to detect a 22% improvement in skin hydration. Okay, so that is why that gotu kola is actually a very common ingredient which actually present in many of the skincare product, cosmeceutical product that you can see in the market today. Okay, back to the core ingredient. One of the core ingredient is called acetococyte. So acetococyte has the anti-inflammatory activity. So how does this acetococyte play the role in inhibiting the inflammation on the skin? Is by inhibiting the cyclooxygenase and the lipooxygenase activity, and they will be able to inhibit the pro-inflammatory cytokine in the skin disease. And once the anti-inflammatory activity happen, we notice that there is a reduction in erythema happens on the skin. And also, patient which has already been treated with extract with acetococyte, there is an improvement in the skin barrier function. And also, other than acetococyte, there is a presence of saponin, flavonoid, and phenolic acid present in the gotu kola. So these few ingredients, basically, they do play a very important role to have a antioxidant and anti-inflammatory activity. So because they have the antioxidant and anti-inflammatory activity, when these two activity act on the skin, they are able to improve the skin barrier function. So when the skin barrier function improve, what we notice is there is a decrease in transepidermal water loss and also the pH value. Just to highlight here that the importance of having a balanced pH value is because based on the literature, there is study actually proven that the more closer your product to neutral pH, there is a higher chances that the bacteria will start to colonize on your skin. This is why that all those skin product that we try to achieve is by having a lower skin pH value, not too low, not until very acidic. So the pH value of skin is mostly 5.5. So the ideal pH value for skin product is about between 5.5 to 5.6. Okay, not too high, because when your product is having a too high pH value, you will actually promoting the colonization of the microbial substance on the skin. And other than that, this saponin, flavonoid, and phenolic acid, they are able to chelate the metal ion. They form the metal ion complex by having a binding with them. And this complex will be able to inhibit the activity of the enzyme and exhibit the anti-lipogenase activity, and eventually that will modulate the oxidative skin damages. Okay, so this diagram actually shows that how acetic acid, which is present in gotu kola, can help in improving the skin condition. Okay, so basically it can be divided into different phases. So this phase is mostly on the anti-inflammatory, which is being played by the acetic acid. Okay, and then after that, we notice that acetic acid, which is present in the Centella asiatica, they are able to promote the fibroblast proliferation, collagen synthesis, extracellular matrices reorganization, angiogenesis, granulation of tissue formation, and also epithelialization. Okay, and this will actually promote the wound closure at the end. So like what I mentioned, because of the excessive scratching that we have, there is a potential that patient with skin disease will have an open wound. So by having this activity in in the treatment, okay, it is able to promote the wound closure. So when the wound is being closed, there is a chances of having infection will be much lower. Okay, so acetocoxide, we also managed to find out that there is literature that actually reported that it is able to decrease the growth of bacteria in the wound surface. However, when we are, my whole entire team, when we started to look into more literature that reported on the effectiveness of Gotu-Kola in inhibiting bacteria, okay, how effective they are in inhibiting the bacteria, especially bacteria which is actually present on skin, we noticed that there is a very limited study reported on the microbial activity. Okay, I think this is the gap that researcher can look into it and see that whether is there any other potential plant extracts or which you are able to actually put together with the Gotu-Kola in order to achieve a better complementary and alternative therapy in managing the skin diseases. Okay, so just to highlight to you that this is what we have been doing in my team, that I have a student who's actually formulating products for the research. So this is a prototype that we have already been produced. Okay, so this prototype is called Axilla Gotu-Kola Moisturizing Cream. So the unique feature that we have in this product is we are actually using the odorless oil. Okay, because some of the plant oil actually consists of odor that some people may not be very favorable to those kind of odors from the plant. So that's why we actually replace with the odorless oil. Other than that, we are using a skin-friendly oil. We notice that there are many moisturizer in the market nowadays actually consists of a specific oil that is actually not very skin-friendly, which is already been proven by the research study. So in this case, we are trying to solve this issue by replacing it with a skin-friendly oil. Okay, and other than that, the Gotu-Kola extract that we are using is actually the oil-soluble extract. The reason why we opt for an oil-soluble extract is for the better permeability, because we notice that compounds which is oil-soluble, they will be able to penetrate into the skin layer and the stratum corneum layer better. Okay, so that's why we opt for an oil-soluble extract. And other than that, we are also using ingredient which is free from nut allergen. Even though nut allergen in terms of topical application is not a very much debated issue, but there are people who actually allergic to protein in the nuts, that we wanted to make our product to be user-friendly to everyone, from infants to older people. So this is why the consideration that we want to take account when we are formulating this product. Okay, so as a conclusion, okay, so basically, I believe that different countries, you have the different kind of traditional medicine kind of application. But what my research team is doing today is that we are using the knowledge that we have from the traditional, from our grandparents, okay, and then try to use the scientific method to implement it into a daily life, that so that whatever product that we wanted to develop or whatever product which is available in the market, there is a scientific evidence to prove that this is effective. Not just like, oh, you say that this is effective, but there is no scientific evidence to prove it. So our team is interested to moving towards the scientific evidence kind of product development in that direction. So tropical plant, it is very commonly used to manage skin disorder. I think if you refer back to your own country tropical plant list, you will be able to find a list of tropical plants which is actually very commonly being used in managing skin disorder traditionally. So when this kind of tropical plants or the finished plant products has already been proven to be effective, this can also be a complementary and alternative therapy. Okay, I'm not trying to say that we try to abandon the first-line therapy and then use this plant product as a treatment. So I will still wanted to say that if your doctor recommend you to use a certain product from the Western medicine to manage your skin disease, you can still continue to use it. but I would want to use some of the plant products as a complementary, or even like if you do not want to use those prescription which is given by your doctor, you can use this as an alternative therapy. And other than that, also plant products, it is commonly popular among individuals. And even nowadays, we notice that a lot of the primary healthcare providers and traditional practitioners, they are actually they quite receptive towards the using the plant products in the as an alternative or complementary treatment. And also, what I want to promote is that this usage of the tropical plant or the finished plant product should play a major role in the integrative medicine, so that it is able to improve the health and quality life of the patients together with the modern treatment. Okay, so finally, I would like to acknowledge a few party who is actually contributing to my research until today. So first of all is our Ministry of Higher Education Malaysia, which actually provided us a fund to continue our research on the pharmacological aspect of khasiat alata. And also not to forget our Research Management Office, okay, SERVI from UCSI University, who is actually continuous funding us on the research so that our study is able to continue for continue to in many years. And other than that, I would also like to thank my collaborator from Monash University, University Putra Malaysia, University of Cyberjaya, and also University Tunku Abdul Rahman. Okay, so these are the references that I have used in my webinar today. And thank you for your attention.
Over to you, Dr. Payal. Over to you, Dr. Payal. Just a minute. Just. Might be there is some technical risk. I would like to take some questions regarding our webinar, ma'am. So from the audience, thank you for sharing your wonderful session. So some of the questions like what are some of the most promising tropical plants currently being researched for their skin diseases therapeutic properties? Can you explain this, ma'am?
Come again?
Just a minute. What are some of the most promising tropical plants currently being researched for their skin diseases therapeutic properties?
Okay, so basically there are different kind of groups. They are focusing on different kind of diseases. What I noticed that I'm talking about in Malaysia. In Malaysia, actually we did a study that to find out that how many percent of the people in Malaysia is actually having skin diseases, and what are the common skin diseases actually found in Malaysia. What we noticed that atopic dermatitis actually takes up about 20% of the people in Malaysia, from infants to about 7 years old. The reason why we focus on the infants to 7 years old is because that during this period that the children is having the much weaker immune system. So, and then this is where they are having the flares of the dermatitis at that age. But eventually, after they have grown up above 7 years old, when the immunity builds up, then eventually there was actually started to have a stronger immunity, then the skin diseases prevalence were actually getting lower. But of course, not to say that children who are actually more than 7 years old, they do not have any diseases. They do have. But in Malaysia, dermatitis is like up to 20%. And other than dermatitis, of course, we do have other issues like psoriasis. We also have dandruff issue. Okay, and of course, actually, I do come on a study on the different kind of diseases, the ranking of different kind of skin diseases from different country. Like example, I still remember that Maldives, actually you have a number one of issue in skin is the dermatitis. Okay, of course, different country you have different kind of ranking. Okay, in Malaysia, that dermatitis is still not the number one, but what I noticed that I was trying to help in improving the lifestyle of the younger generation, because I noticed that there's a lot of parents, they are unwillingly to use a steroid for their children, even though that the skin condition has already become very serious. But whenever they go and see the doctor, they actually very hesitate to use the steroid for their children. So in this case, there are different kind of plants which is available, okay, in Malaysia to handle the skin diseases. So among those plants which are present in Malaysia, these are the 3 top ones which actually being commonly used in Malaysia. Not to say that only these 3. We have a list of different kind of plants which is being used by traditional medical practitioners, but these are the 3 that actually I wanted to bring up to the attention of everyone, because some of the plant, it could be commonly present in Malaysia, but not in other part of the country. So I believe that like coconut oil, I think everyone will know what is coconut oil. Okay, kasih alata, maybe some of the country you know what is it, some of it you may not know. Okay, and also Kuala, I think this is also a quite common candidate in managing the skin diseases. So there's a very long list of those plants which is available to handle skin diseases.
Thank you, ma'am, for this wonderful answer. So my next question is, how do environmental factors influence the medicinal properties of the tropical plants, and how can these be controlled or optimized?
How does the environmental factor affect the
Influence the medicinal properties of the tropical plants?
environmental? Correct me if I have a correct understanding that how does the environmental factor affect the medicinal property of the tropical
plants, right?
Mm,
And how can we, if they really affect, how we can optimize those conditions, such conditions?
okay, plants, basically, if you ask me that different places, okay, different places, even though you are in the same country, when the growing condition is different, based on our previous research all this while, we noticed that plant source, when you actually get it from the same country, but it's from different location, the properties or even the phytochemical content which is present inside there, it can differ a lot. Even not only that, from the different places, what we noticed that the period when we harvest those materials, the plant materials, is different. Like example, maybe one samples I did it during January, and then one sample I did it maybe in April or even May, which is a lot of rainfall. we notice that when we try to do the analysis of the phytochemical composition, that differs a lot. So it depends on the condition which is actually present in the growing environment. So that's why that when this is also one of the challenges that we have to solve when we are trying to get a very stable source of the plant material, that we can make sure that the phytochemical composition is always the same. That is also a very challenging part. So this is why that part of my research team, instead of using the very conventional way of like go and collect the plants, dry it, and process ourselves, we actually have a team that is actually doing standardized extract, which means that we will only use the extracts which is already been standardized commercially, so that we know that every batch of this extract will be stable. Okay, and then the result that we do, it is reproducible. So that is also one of the strategy that I have two different kind of teams is looking at.
Okay, thank you, ma'am. The next question is, what are the challenges in integrating the herbal medicine into the mainstream dermatological practice?
Okay, in Malaysia, okay, basically we still have like different kind of races of traditional medicine practice. Okay, the receptiveness of the people in Malaysia is not so much that they are very willingly to accept that using plant products. Okay, but the only thing is what their concern is that whether the product that they are using is free from certain drugs. Like example, very commonly people will ask about whether is this free from steroid. Okay, because especially there are products that after the application, they notice that the result is fantastic. Only 1 to 2 application, the result is fantastic. Then people will start to question, is this a steroid-free product? I know that some of the users that actually took the samples and then sent out to the commercial lab for the analysis to try to find out, is this a steroid-free? But of course, having a steroid-free product is always the number one concern. Okay. Other than that, there are another challenges that we also face in Malaysia is there are a lot of products that is actually not being registered. Okay, that means that it can be put online and then people have been writing some review. Oh, this product is very good, and then it is they don't have a sort of like product registration with the relevant body in Malaysia. So that is also one of the challenges that we need to educate to our patients, that whenever that what kind of product that you use, you must make sure that this product has already been tested and this is safe to be used. Not to say that, okay, you just based on the review that you actually read online and then you just believe as it is. Okay, so that is a safety part that we need to educate our consumer.
Thank you, ma'am. The next question is, what are the safety considerations and potential side effects when using the plant-based therapies for skin conditions?
Okay, plants, because what we are in plant products, what we have been using is we know that there is a mixture of phytochemical compounds inside. Some people, you may be allergic to certain phytochemicals, but not everyone. So that's why in this case, whenever that when you are using a new products, that means that you are first time using the new products, it will be better for you to test out whether are you allergic to the ingredient inside. Other than reading the ingredient from the product description, you can also like have a small surface area to do the testing on the irritation. I think this is number one that everyone must do before you started to like put the phrase on your cream. Put the cream on your face and then started to have a very big area of that. Then only you find out that you're actually allergic to one of the ingredients inside. So it is always best to do the testing, see whether are you allergic to the ingredient inside. If no, then only you started to apply to other parts of the body. So that is a safer side that we also need to constantly educate our patient.
Okay, ma'am. The next question is, ma'am, what regulatory hurdles exist for the approval and commercialization of the plant-based skin products?
Okay, different country, you have a different regulation for that. Like example, in Malaysia, we have a regulation on if your product is not towards the drug category, okay, the regulation will be actually much more easier, okay, because they will only having a certain specific test that you need to fulfill to prove that this product is safe, like example, like heavy metal, your microbial count, okay, and then basically those are the simple information that is required in the terms of registration for the products. But if your product is going towards for like therapeutic purpose, like example that you are trying to push it as a drug product to handle the skin diseases, the regulation will definitely be much more tougher. Okay, I do believe that different country, whether you are from Malaysia, you're from Singapore, from Brunei, from India, okay, from Maldives, the kind of regulation that you have to go through, it will be different because this is subjected to your own country regulation, that what kind of criteria that you must fulfill before you can sell the product in the market.
Okay, ma'am. Ma'am, can you share some success stories where tropical-based plant therapies have been significantly improved patient outcomes in the skin disease management?
Okay, just to share one of it, which is actually I did not put it in the slide today because today I was actually very focusing on dermatitis. Actually, my team, we did a product development from scratch until the commercialization part, that we actually use a standardized extracts and try to formulate it into a washing product, okay? So it's a shampoo, basically. So we are actually incorporating only the natural and natural derived ingredient in our product. And we put in the standardized extract from a plant, which actually with the proven kind of literature activity. And then after we put it into our product formulation, we did the testing in vitro to see that how effective is this product on handling the fungus. So because this is an anti-dandruff product, so we're actually trying to see that how well it actually inhibits the dandruff fungal. So we found that, okay, at certain percentage, we noticed that there is an optimum percentage of the standardized extract that we need to put in. And then we moved to the clinical trial. And of course, we put our patients subjected to a 6 weeks clinical trial. And every week, the patient will have to come back to follow up with us. Then that is where we will actually observe the improvement of the hair scalp, which actually see that the improvement of the whole entire dandruff issue. And we noticed that majority, up to 80% of them, they actually mentioned that the product actually works. Okay, so that is what actually makes our whole entire team so confident to have this product to launch to the market. So actually the whole entire commercialization process is already in the pipeline.
Okay, thank you, ma'am. Next question is one of the person has asked, can fatty acid derivatives with any heterocyclic scaffold helpful in skin diseases?
This one, I'm not too sure on what kind of free fatty acid that you mentioned. What I can say that in the coconut oil, there is actually a literature that have already reported that the conversion of this medium chain fatty acid into a free fatty acid, this free fatty acid is the one that actually inhibit the S. aureus from growing. Actually, it's not the medium chain fatty acid. Okay, if you just put in the medium chain fatty acid and you try to inhibit those skin microbial from growing, there is nothing actually promising that. And then after the conversion of this medium chain fatty acid to free fatty acid by the skin microflora, then notice that that is where the whole entire thing started to work. So that's why that sometimes it's not that the product that the compound that actually we put in play the role, is after the conversion by the skin microbiome actually is the one that actually play a very major role in that.
So one of the person has asked a question that, is it the formulation or the excipient that affect the efficacy of the product?
Okay, one thing is in our study, we did test on the product without the extract and product with the extract, and we are able to see the comparison. Of course, no doubt that product without the extract, we do have some excipient because like what I mentioned, that the excipient that we are using is plant-based or plant-derived. Okay, so if you are using this kind of ingredient, definitely we will not deny that there is some kind of activity is actually present there. But when after we do the comparison, we notice that with the addition of the standardized extract, the activity becomes greater. Okay, even though in the base there is some activity, but it's very mild. It's not good enough to have a very significant result. But after the addition of the core ingredient, that the activity actually goes much higher. So that is why we are very confident that our product with the standardized extract is the one that is actually playing the effective role in inhibiting in exhibiting the activity.
One of the question is, ma'am, can you please elaborate about the wound healing process with respect to the dermatological products?
Okay, so basically for wound healing, most of the time for whatever research that you wanted to do on wound healing, first of all, you always go into in vitro study. And in vitro study, most of the time, we are using the cell lines to do. And if you look at the literature, it is very common that the scratch assay is a common assay that these people have been using. Okay, so if you're not sure that what is scratch assay, let me elaborate briefly to you that basically you are using one kind of cell line. Okay, you try to grow them on the well, on the micro well. Then after that, you do a scratching in between. Okay, you treat the cell, and you will observe whether the cell will be able to have a closure for that. Okay, throughout the period of time. Some of the extract, if you have the wound healing activity, the wound that the scratch that you created, you will notice that it will have a closure. But if plants that do not have the wound healing activity, you will notice that the cell is either they don't grow or they will just slowly die off. Okay, so this is usually the very preliminary study that people will be using. Secondly, if in the in vitro assay, you are confident that your extract do have the wound healing activity, the next phase people will usually go into the in vivo study, which is using animal. So by using animal, you will be able to see that how this whole entire thing works. Okay, so of course, it depends on what kind of animal that you are using. Different people, they are using different kind of animal. Some of them, they're using mice, some of them using rats, some of them using even until rabbit. Okay, I have seen different kind of studies before. So basically, the essence of having the in vivo wound healing is to you create a wound on the animal. Okay, and this wound, you will try to observe whether this wound it will close up after how many days, and then you try to compare with the control. So if you notice that the wound healing activity is actually much better than the control, that means that your plant extract actually works as a wound healing. Okay, if you notice that the wound closure is not satisfactory, okay, then you can say that your wound closure or wound healing activity is not satisfactory. Okay, so I do believe that probably you may have heard from your grandparents that you can use certain plants and then you try to make it into paste, put on your wound, and then just leave it for some time. That is the traditional knowledge that our grandparents is actually giving us. But for us as a researcher, as a scientist, we are trying to implement this knowledge into a more scientific way.
Thank you, ma'am. One last question is, what are the future directions and the potential research areas in the field of tropical plant-based dermatology?
Okay, so like what I have been doing now, okay, I think the future direction is that on the plant-based therapy is not to replace the first-line therapy. Okay, first-line therapy is always the major preferred therapy. What I was trying to push is that probably we can actually use a plant-based therapy as an alternative, which means that if let's say you have only a very minor skin issue, you may not want to use the first-time therapy, but you think that it's too harsh for your skin, you can go for alternative therapy first, like example, using plant-based products. Okay. Alternatively, there are people who actually having a very serious skin issue, which I've seen, that they will just continue with the first-time therapy, but they use this as a complementary therapy to help out with the whole entire process. Because, like example, topical corticosteroid, they do have the side effect. Okay, you can't use topical corticosteroid for long term. Okay, you need to stop at certain point, because the longer that you use, when you are having dependency on the topical corticosteroid, that is where you cannot stop using this product. And once you stop using the product, you will notice that there's even a much serious flag after that, which is actually, I noticed that there's a lot of people is actually having, because due to the lack of education, that they just keep continue using it, even though the skin heals, they should stop. They did not stop. Okay, they just continue until that they realize that one day when they wanted to stop, the whole entire skin flare is even much serious than previously. That is the common steroid withdrawal that they have to face. Okay, so I would suggest that if you are having a very serious skin issue, you can use the first-line therapy, which is recommended by your physician. And then at the same time, you use plant-based product as a complementary therapy. So when you started to lower down the dosage of the first-line therapy, because you are still continue using the plant-based for the maintenance. So that's why I don't promote people that, oh, you should stop the conventional therapy that your physician is giving and then use the plant-based. I will not promote that. I will usually say that you can use that as a maintenance for long term, because we know that exposure to those Western medicine for long term, it may not be beneficial to us. So eventually, to sometime we may need to look for alternative that you can use it for long term management.
Okay, ma'am, one more question has been asked by the audience because of that wound healing topic, that question you have discussed. Based on that, that is formulation of herbal film forming spray is possible for wound healing or various skin diseases?
Is the formulation for herbal base?
Herbal film forming spray.
Oh, that
Spray
depends.
is here that is forming a herbal film.
I
Is it possible for wound healing?
I do see some of the study, they are trying to put the herbal ingredient onto maybe like hydrogel or even like foam. Okay, and then they try to put it there and then see whether it's effective or not. I would say that it depends on the design of the experiment, of course, of also the ingredient, whether the herbal-based ingredient that you actually put on the foam is able to penetrate into your skin or not. If your ingredient is just remain on the surface of your skin and there is no penetration, I do believe that you may not notice that it is effective. So there is a lot of different consideration that you may want to look into. For example, if you notice that the penetration of the active ingredient is not good, then maybe you want to like formulate into smaller particles, or even to improve the delivery method into the topical layer. So that is where you have to look at how the design of your whole experiment. Okay.
Okay. Thank you so much, ma'am, for your valuable insights and a wonderful
My
topic,
pleasure. My
interesting
pleasure.
one. So, as we conclude our 52nd SPSR International Webinar on Nature's Pharmacy, Tropical-Based Plants Therapies for Skin Disease Management, I would like to extend my deepest gratitude to all our distinguished speakers, participants, and organizers who have made this event a resounding success. Today's discussions have underscored the immense potential of tropical-based therapies in revolutionizing skin disease management. We explored the rich phytochemical properties of these plants and their significant antioxidant, anti-inflammatory, and antimicrobial activities. The insights shared have not only enhanced our understanding, but also have opened new avenues for the development of natural, effective, and safer treatments for various dermatological conditions. The pioneering research, particularly in the identifying plant-derived compounds for therapeutic use, showcases the profound impact nature has on modern medicine. Integrating these natural remedies into the contemporary skincare practices promises a holistic approach to health and wellness, emphasizing the importance of sustainable and eco-friendly solutions. So, as we move forward, let us continue to collaborate, innovate, and explore the vast potential and nature's pharmacy hold. By combining traditional knowledge with modern scientific advancements, we can develop groundbreaking therapies that benefit patients globally. We extend our sincere gratitude to Dr. Shashi Alok, the National President of SPSR, for gracing us with this esteemed presence today. In acknowledging your dedication and leadership, we express our heartfelt thanks for your valuable contributions. In extending our deepest gratitude, we express our sincere appreciation to the esteemed speaker, Dr. Chew Yik-Ling, whose generosity in sharing his profound expertise and valuable experience as well as perspectives has enriched our understanding of nature's pharmacy, tropical-based therapies for skin disease management. As special acknowledgement is extended to our convener, Mrs. Monika Sabharwal, whose pivotal role in orchestrating this insightful event has been instrumental. We extend heartfelt thanks to all the participants, recognizing the collective effort that has effectively pushed the boundaries of knowledge and fostered a spirit of collaborative learning. Together, your contributions have made this webinar a resounding success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. Thank you for being an integral part of this 52nd SPSR International webinar. Your active participation and contributions have been invaluable, and we look forward to future endeavors that contribute to the continuous betterment of healthcare and well-being for all. Thank you once again for your participation. Have a wonderful day ahead. Thank you, ma'am.
Okay, thank you, everyone.
We may leave, ma'am. No.